RNA polymerase 2 will synthesize messenger RNA. Messenger RNA. RNA polymerase 3 will synthesize transfer RNA. So for you to remember it's RNP. Polymerase 1R ribosomal. Polymerase 2N messenger. Polymerase 3T transfer RNA. So those are the enzymes involved in the synthesis of RNA. So step one, for transcription to occur, the double helix must unwind. When the double helix will unwind now, what will happen? So what strands are produced during unwinding of DNA? So one, you have the informational strand. Because DNA is composed of two strands. When it will unwind, then two strands are formed, and this one is called the informational strand. While the other strand is called the template strand. So one, you have a template strand, and the other, the informational strand. Now, which strand is used for RNA synthesis? Which strand is used for RNA synthesis? So let's go back. RNA is composed of one strand only. So that's the reason why only one strand of the DNA will be utilized in the synthesis of RNA. And the strand in the DNA molecule that is utilized in the synthesis of RNA is the template strand, not the informational strand. So the strand utilized in RNA synthesis, this one. This is the template strand. So this is your DNA molecule composed of two strands. They will unwind. This strand running from the 3' to the 5' direction. This is the template strand utilized in the synthesis of RNA. While this one is the informational strand. So therefore, in the synthesis of mRNA, it is dictated by the sequence of nucleotides coming in 3 in the template strand. So what did we say in the pairing of bases? So if this is guanine in the template strand, then in the mRNA, it becomes cytosine, your guanine. Cytosine becomes guanine, adenine becomes uracil, thymine in the template strand will become adenosine. Okay? So, what is the effect of the unwinding of the DNA double helix? It will now expose the bases in the template strand. So, what is the effect of the unwinding of the DNA double helix? If this is the template strand running from the 3' to the 5' end, these bases are now exposed. And you have nucleotides in sequence acting as the template in the synthesis of RNA. So the bases in the template strand becomes exposed due to the unwinding of the double helix, and in the template strand, the bases there will serve as a template in the synthesis of RNA. That's why the strand utilized in the synthesis of RNA is called the template strand, because the bases found in it will act as a template in the synthesis of the RNA molecule. So next, when the RNA synthesis will start, first it is important that the RNA polymerase will be able to recognize the ionization point. So when will transcription process begin? When RNA polymerase is able to recognize the initiation point. And the initiation point in the template strand is called the control segment. It's also called the control segment or the initiation sequence. Again, it's called the control segment or the initiation sequence. So what is this sequence? It is composed of 3 nucleotides, 1, 2, 3. So that is the initiation sequence. Okay? And if it is recognized by the RNA polymerase, then it will signal the start of the synthesis of RNA from the initiation sequence or from the control segment. So the blue colored one, this is the template strand, and RNA polymerase will be able to recognize this initiation sequence. And that's when transcription will begin. Next. So step 2 will be the assembly and elongation. This is step 2. The assembly and the elongation of the RNA, whether it is mRNA, RNA, or transfer RNA. So in what direction does RNA polymerase move during RNA synthesis? So you have to be guided by this blue color template. So the RNA polymerase will move from the 3' to the 5' end of the template strand. The RNA polymerase will move from the 3' to the 5' end of the template strand. Okay, let's go back so that we can see it. So this is the 3' to the 5' end. This is the 3' to the 5' end. So this RNA polymerase will move, starting here, moving down, until there is a termination sequence. This is a termination sequence. In what direction does RNA synthesis proceed? If this is the template strand running from the 3' to the 5' end, an RNA polymerase will move from the 3' to the 5' end, then synthesis of RNA will proceed from the 5' end to the 3' end. Opposite in direction. The synthesis of RNA using the template strand. RNA polymerase will move from the 3' to the 5' end, while the synthesis of the RNA molecule will proceed from the 5' to the 3' end. So during assembly and elongation of the RNA, what determines the RNA nucleotides that will be added to the growing RNA chain? It will be determined by the sequence of the bases. It will be determined by the sequence of the bases found on the template strand. So what determines the RNA nucleotides utilized in the synthesis of the RNA? What did we say? RNA is composed of ribonucleotides. So therefore, what determines the ribonucleotides that will be utilized in the synthesis of the RNA molecule? It will be determined by the sequence of bases found in the template strand. Okay. So you have triplets of bases coming in phase that will now dictate what ribonucleotides will be added to the growing, this one, to the growing RNA molecule. Okay. So this is the base pairing during synthesis of RNA. Do not be confused. I'm just using this DNA molecule during replication because I cannot find an a template DNA strand that dictates the synthesis of an RNA molecule. So for example, this is this. So for example, this is now the template DNA strand utilized in the synthesis of an RNA molecule. If this is T, then in the growing mRNA or RNA or tRNA molecules, thymine pairs with adenine. So what the ribonucleotide on the mRNA, growing mRNA, will be adenine. If it is cytosine, it pairs with guanine. If it is adenine, so the next base here in the mRNA should be uracil. It should be uracil. So that's complementary base pairing in the RNA, in the synthesis of RNA molecule. So let's go back. What determines the RNA nucleotides? Again, we have said the sequence of bases based on complementary base pairing. According to complementary base pairing. So when will termination of transcription will occur? When the moving RNA molecule moving down the 3' to the 5', if it will now recognize a termination sequence. A termination sequence. So again, a termination sequence, like the initiation sequence, it is composed of 3 nucleotides. And the termination sequence will now signal the termination of the synthesis of your RNA molecule. It will now signal the termination of the synthesis, this one, of the RNA molecule. So what happens at the end of transcription? After RNA polymerase will recognize a termination sequence. The RNA synthesized will now be released. And the second thing that will happen, the DNA will rewind to form back the original DNA molecule. So this template informational strand again will rewind. So you have now the DNA strands rewinding to regain their structure. And RNA synthesized becomes released. So that's what will happen at the end of transcription. Questions so far? So next, after mRNA is synthesized or any type of RNA, what will happen? The RNA produced is not functional in nature. So that's the reason why there is what we call post-transcriptional modification of the RNA synthesized. What's post-transcriptional? Post, after synthesis of the RNA, it will be modified so that it becomes functional. So the name of the RNA produced during transcription is called heterogeneous. Okay, nuclear RNA. So heterogeneous RNA. Or the other name, if it is an mRNA molecule, it's called a primary transcript. The other name for the heterogeneous RNA synthesized during transcription is also called a primary transcript. So what will happen during post-transcriptional modification? So let's go back. During synthesis of RNA from the template strand, there are regions in the genes found in the DNA strand that are coding and non-coding. Okay. So what is an exon? An exon, colored green, are segments in the gene that codes for the synthesis of an amino acid during protein synthesis. So the importance of synthesizing mRNA now will dictate the synthesis of the protein molecule in the ribosome. So during synthesis of mRNA, what happens? Both the coding and the non-coding segments of the DNA are being copied during transcription. So exon, this is the coding segment in the gene that will code for the synthesis of an amino acid. While introns, colored violet, these are non-coding portion. These are non-coding segments of the genes. They do not code for the synthesis of an amino acid. So that's the reason why during post-transcriptional modification of RNA, the introns will be removed. The introns will be removed. So it will be cut, the introns, colored violet. And after they are removed, these exons are joined together. These exons are joined together. So the introns are excised. And after the introns are excised, then the exons are joined together. And what will do that? A protein we call splesiosome. This is the spelling of splesiosome. It will be a protein, a complex of protein molecules called splesiosome. So the process of removing introns, excising them, and joining together exons is called splicing. S-P-L-I-C-I-N-G. Splicing. And the protein complex that will do splicing, it is called splesiosome. Okay? So after the introns are removed, the initial mRNA synthesized called the primary transcript or the heterogeneous RNA, it becomes now functional mRNA. It becomes functional mRNA. Why is it that the primary transcript or the heterogeneous RNA is not functional after its synthesis? Because there are intervening introns in the sequence that are non-coding. To make it functional, the introns which are non-coding segments, they need to be removed. So that during synthesis of the protein, the sequence of the amino acid will come one after another. So that's post-transcriptional modification of the RNA that is produced. So splicing is the process of excising introns and joining together exons. And the molecular complex that accompanies splicing is called your splesiosome. So this is an example, your alpha globin and beta globin gene families. It contains 3 exons, okay, and 2 introns that become removed by splicing. So when the mRNA now is functional, it can now code, for example, for the synthesis of an alpha and a beta globin chain in your hemoglobin molecule. So the alpha globin and the beta globin genes are the ones that are being utilized in the synthesis of mRNA using the template strand. And when an mRNA is formed now, then it will now dictate the synthesis of the polypeptide chains in your hemoglobin molecule. So we'll now move to another topic, genetic code. So what is a genetic code? A genetic code refers to the sequence of nucleotides coming in threes in your messenger RNA. Again, the genetic code refers to the sequence of nucleotides coming in threes in your messenger RNA, which will dictate the synthesis of proteins in the ribosome, which will dictate the synthesis of proteins in the ribosomes. And a triplet of a nucleotide in your mRNA, we call it a codon. We call it a codon. So what is a codon? A codon is a triplet of nucleotides. When we say triplet, 3 nucleotides. So a codon, in other terms, it is being defined as a sequence of 3 consecutive nucleotides found in the mRNA molecule that will dictate the synthesis of proteins. For example, in this messenger RNA, okay? So if this is the codon found in the messenger RNA, GCU, glutamine, cytosine, uracil, will code for the synthesis of alanine. Okay? So that is a codon, a triplet of nucleotides that will code for the synthesis of a specific amino acid in the polypeptide chain. So how are codons written? How are codons written? It is written starting from the 5' end to the 3' end. So again, what did we say earlier? During synthesis of RNA, RNA polymerase will run from the 3' to the 5' direction. And the direction of the growing mRNA will run from the 5' to the 3' end. So codons are written starting from the 5' to the 3' end. Okay? The codon at the 5' end will now determine the amino acid found in the N terminal of the polypeptide chain. So this is a peptide chain. How many amino acids? 1, 2, 3, 4, 5, 6. So you have a hexapeptide. And the first amino acid in the hexapeptide at the 5' end is the amino acid found at the N terminal, meaning N, the amino terminal. While the last amino acid found at the 3' end is the amino acid at the C terminal or the carboxyl terminal. So if this is the messenger RNA, it will now dictate the sequence of the amino acids synthesized or found in the peptide chain that is synthesized. So for a protein to be functional, it must have the correct sequence of amino acids in its polypeptide chain. If you can remember our case presentation on sickle cell anemia, any mutation causing a change in the sequence of these nucleotides will change the amino acid at a particular position in the peptide chain. For example, in sickle cell anemia, there is a mutation in the gene that calls for the synthesis of glutamine. At the 6th position, it becomes replaced with valine. And therefore, it will now affect the structure of the hemoglobin molecule and its function, that of transporting oxygen molecules. So it is very important the sequence of the amino acids in the peptide chain, because it will determine how the peptide chain will fold and assume a tertiary or quaternary structure, its native cone formation, so that the synthesized protein will be able to carry out its function. Discuss how many possible combinations of nucleotides are there that can code for the synthesis of the 20 different amino acids. Now, in an mRNA molecule, how many nucleotides are there that are utilized in the synthesis of the mRNA molecule? How many nucleotides? 8. Okay, very good. There are only 4 nucleotides. Okay, let's go back so that you will understand this one. There are only 4 nucleotides found in an mRNA molecule. One nucleotide is thymine, another cytosine, another adenine, and another uracil. Thymine and adenine, cytosine, and guanine. Okay? So how many? 4 only. What are they?
Adenine, cytosine, guanine, uracil.
So there are only 4. Take note, there are only 4. Adenine, guanine, thymine, uracil. But there are only 4. Okay? So what will happen now? If there are 4 nucleotides in an mRNA molecule, how many nucleotides are there in its codon? How many nucleotides are there in its codon found in the mRNA molecule? There are only 3. So therefore, how many possible combinations of nucleotides will be there? 4 nucleotides utilized in the synthesis to the 3rd power, because you have only 3 nucleotides in the codon. So 4 times 4 equals 16, times 4 equals 64 possible combinations of nucleotides. So these are the 64 possible combinations. Let's count. In its big box, you have 1, 2, 3, 4. Okay? And then 5, 6, 7, 8. 8 plus 4, 12, 16. 16 times 4 nucleotides. So 16 times 4 becomes 64. So there are 64 possible combinations of nucleotides coming in threes. Each codon in the messenger RNA. So these are the different codons coding for a specific amino acids. So what information does this table give you? Therefore, several codons can code for one specific amino acid, while only one codon will specify the synthesis of methionine and tryptophan. 2 will specify the synthesis of tyrosine. These 2 codons. 3, are there 3 here? 3 for isoleucine. 4 for proline, and so on. So the one with the most number of codons specifying for its synthesis, there are 6. 1, 2, 3, 4, 5, 6 for arginine, for leucine, and for serine. Okay, so these are the number of codons coding for the synthesis of a specific amino acid. So how many codons code for specific amino acids? There are 61. There are 61 codons that code for a specific amino acid because these 3 codons are stop codons. So out of the 64, there are 3 stop codons. What is the meaning of the stop codon? Okay, very good. It signifies termination of protein synthesis or termination of a polypeptide chain elongation during protein synthesis dictated by mRNA in the ribosomes. So remember these 3 stop codons, UAA, UAG, and UGA. These are the 3 stop codons. The rest of the codons are coding codons, meaning they code for a synthesis of amino acid, while stop codons are non-coding codons. They do not code for the synthesis of a specific amino acid, but they will signal termination of synthesis. Question? Yes, EUJ is the only initiation codon. When EUG is found as the start at the 5' end of the messenger RNA, it will signal initiation of synthesis of a polypeptide chain. Just like in the synthesis of an mRNA, there you have 3 nucleotides that acts as a signal for the initiation of the synthesis of the mRNA, and the synthesis of a polypeptide chain, EUG will act as a signal for the start of the synthesis of a polypeptide chain. If EUG is found anywhere in the mRNA molecule except at the first, then it will code for the synthesis of methionine. When it is found as the first codon in the mRNA molecule, automatic methionine will be synthesized also. But during post-transcriptional modification of the protein, the methionine will be removed also. Otherwise, all protein will start with methionine, and that cannot be possible because different proteins will start with different amino acids. Each protein is a unique molecule, having its own specific sequence of amino acids. Okay. So what is the initiation codon? It is the codon that will signal initiation of protein or polypeptide chain synthesis, and that is your AUG. What is its role during protein synthesis? It signals initiation of synthesis of a polypeptide chain in the ribosome. What is the use of codons that do not correspond to any amino acid? So these are what we call termination or stop codons. They signal termination of synthesis of the polypeptide chain. What are termination codons? They signal termination of the synthesis. We'll now discuss the characteristics of the genetic code. Remember the genetic code? This refers to the sequence of nucleotides coming in threes in the messenger RNA that will dictate the synthesis of protein in the ribosome. Its first characteristic, it is universal. What is the meaning of that? That these different codons will code for the synthesis of amino acids in every organism, whether it is a plant, an animal, a human being. If three U's will code for phenylalanine in the human being, it's the same codon that will dictate synthesis of phenylalanine in plants, in animals, and so on. So therefore, the genetic code is universal. Second, the genetic code is degenerate. What is the meaning of the genetic code being degenerate? Several codons can code for a specific amino acid. That's the meaning of degeneracy, or a codon being degenerate. Several, more than one codon, to be exact, more than one codon can code for the synthesis of a specific amino acid, except methionine and tryptophan. Degenerate meaning more than one codon can code for the synthesis of a specific amino acid, except methionine and tryptophan. Only one codon codes for tryptophan and methionine. Next, on degenerate. So this is just showing you the different codons coding for these different amino acids. And for tryptophan, there is only one. Where is methionine here? It's not being shown. But methionine is being coded by one codon also. Next, unambiguous. What is the meaning of unambiguous? The genetic code. So when the synthesis starts at the first codon, then what will happen? The reading of the codon will continue from the start to the end. Meaning from the start, when there is an initiation codon, and towards the end, when there is a stop codon. So it is unambiguous. So the other meaning of unambiguous is that when a codon codes for phenylalanine, it will always specify for the synthesis of phenylalanine. So therefore, when we say a genetic code is unambiguous, it codes for a specific amino acid. So each codon will code for a specific amino acid. That's the meaning of unambiguous. Number 4, non-overlapping. So for example, this is now the mRNA, and usually the first will be AUG because that is the initiation codon. So when the mRNA, when the ribosome moves over the mRNA molecule, then it will move reading codons in sets of 3 nucleotides. So this is the first set, 3 of them, the second, the third. So therefore, the reading of the codon is non-overlapping. So the codon are being read in sets of 3 nucleotides. One set, 3, 3, 3. If it is overlapping, after the ribosome will read 3 nucleotides. If it is overlapping, then the next will be UAC, and then ACG. So that's overlapping. But when we say the genetic code is non-overlapping, when it is being read by the ribosome, it comes in threes. And after reading one codon composed of 3 nucleotides, it moves to the next codon composed of another 3 nucleotides and down the line until that stop codon is reached. So it is non-overlapping. Number Number 5, not punctuated. Meaning once the ribosome will read the first codon, it moves from the first going to the last without punctuations or without a comma. So it will just proceed. And what makes this possible? Because the introns have been removed during splicing. So that once the initiation codon is being read, then the ribosome will continue reading until it ends in the stop codon. So not punctuated, it's commaless. So those are the 5 characteristics of the genetic code. So we'll now move to translation, protein synthesis. Any questions so far before we move to translation? What time is it? Okay. We'll just finish translation. Define translation. Translation, to be simple, is just synthesis of protein using mRNA as the template. In protein synthesis, the template being utilized to dictate the synthesis of protein in the ribosome is your mRNA. While in the synthesis of mRNA, the template utilized is the template strand of the DNA molecule. Or in other textbook, translation is being defined as decoding the information found in mRNA. And once it is decoded, then it will now be utilized in the synthesis of a polypeptide chain. State the role played by the different types of RNA during translation. Your mRNA is the template containing the codons, and each codon will dictate the synthesis of a specific amino acid in their correct sequence. So that's the role of mRNA. The ribosome, a component of the ribosome is an rRNA. So it is the site of protein synthesis. So the role played by ribosomal RNA, it is a component of the ribosome, which is the site of protein synthesis. What about your tRNA or transfer RNA? It is the type of RNA that brings the specific amino acid to the ribosome that will be utilized in the synthesis of a polypeptide chain. So it is the transfer RNA that brings the specific amino acid. So what makes it possible for tRNA to bring the correct amino acid during protein synthesis? In a portion in the tRNA molecule, there is what we call an anticodon. In a certain portion of the tRNA molecule, we call the anticodon arm, there is what we call an anticodon that is complementary to the codon in the mRNA. So if the codon in the mRNA is AUG, the anticodon of the tRNA will be U pairing with A, then A pairing with U, and C pairing with G. So therefore, this will enable the tRNA to bring the correct amino acid, which will be, if that is AUG, that will be methionine. Because there is an anticodon in the tRNA molecule. So the tRNA ensures that what it brings, the amino acid it brings to the growing polypeptide chain is the correct amino acid. The RNA cannot determine if it is the correct amino acid that is attached in the growing polypeptide chain. It is the role of the tRNA. Okay. So state the 3 steps in translation. First is initiation. Second is elongation of the polypeptide chain or the peptide chain. And the third is termination. So there are only 3 steps: initiation, elongation, and termination. Okay, we will start with the first step. Before we start with the first step, let's first talk about amino acylation of tRNA. So what is amino acylation of tRNA? There are 2 steps in this amino acylation of tRNA. What is the first step? The first step is the activation of the amino acid. The first step is the activation of the amino acid, specifically the carboxyl portion of the amino acid. How is an amino acid activated? By reacting with a molecule of ATP. How? The alpha carboxyl, this oxygen in the alpha carboxyl group. Remember, this is alpha carboxyl because this is attached to the alpha carbon atom, the chiral carbon atom in your amino acid molecule. So the oxygen atom in the carboxyl group will carry out a nucleophilic attack on the alpha phosphoanhydride bond. And what will happen? This phosphoanhydride bond will become cleaved, and these 2 phosphoryl groups will be released in the form of a pyrophosphate. And this now causes attachment of the amino acid via its carboxyl group to these phosphorus atoms. So what will happen? This will be cleaved here, and this phosphorus atom will accept electrons from this oxygen atom in the carboxyl group. In that way, the amino acid now becomes attached to this what? Adenosine monophosphate. So that's why the product of the activation of the amino acid is called aminoacyl adenosine monophosphate, or 5 prime. Why 5 prime? Because the amino acid is attached to the phosphoryl group, and in turn the phosphoryl group is attached to the 5th carbon atom in the ribose sugar in your adenosine monophosphate. That's why it's called 5 prime aminoacyl adenylate, or aminoacyl adenosine monophosphate. So that is the first step, activation of the amino acid. So what is the importance of the activation of the amino acid? Later, it will facilitate the formation of peptide bond. It will facilitate the activation of the amino acid, will facilitate the formation of the peptide bond during peptide chain synthesis. It will facilitate the formation of the peptide bond during peptide chain synthesis. So that's the importance of the activation of the amino acid. Next, the activated amino acids will now be attached to their specific tRNA. So how are amino acids attached to their specific tRNA? So what will happen now? So this is now your amino acid. Okay, this is the amino acid, adenosine monophosphate. So you have the ribose sugar here. So we look at this next slide to be able to see everything. Okay, sorry. First, let's talk about the importance of attaching the activated amino acid to their specific tRNA. What is the importance of attaching amino acids to their specific tRNA? So there are 20 different amino acids. Therefore, there are 20 tRNAs carrying its amino acid, each of the 20 amino acid. And what is the importance of attaching the amino acid to their specific tRNA? It will ensure that the amino acid being added to the peptide chain is the correct amino acid as dictated by mRNA. So if you can still remember, how will an amino tRNA bring the specific amino acid dictated by the codons in the mRNA? It has an anticodon, the tRNA. I have shown it already. It has an anticodon. So in that way, the attachment of amino acids to their specific tRNA, it becomes specific. So there are, So there are two ways of attaching activated amino acids to their tRNA. So one, the hydroxyl, the oxygen atom in the hydroxyl group in the second carbon atom of the ribose sugar will carry out a nucleophilic attack on this carbonyl carbon. So this now will become attached to the oxygen atom in the second carbon atom of the ribose sugar. And then in the third carbon atom of the ribose sugar, this oxygen again will carry out a nucleophilic attack on this carbon atom. And this carbon, this amino acid will now be transferred to the oxygen atom in the third carbon sugar. Okay? So that is what we call trans-esterification. So first, the oxygen atom of the hydroxyl group in the second carbon atom of the ribose sugar will carry out a nucleophilic attack on the carbonyl carbon, causing an ester bond to be formed. But this ester bond will be transferred here. That's why the process is called trans-esterification. The amino acid is being moved from the second carbon atom of the ribose sugar to the third carbon atom. But in a certain class of enzyme, automatic the oxygen atom in the third carbon atom of the ribose sugar will carry out a nucleophilic attack on this carbonyl carbon, and automatic the activated amino acid is directly attached to the third carbon atom of this ribose sugar. So that is how an activated amino acid becomes attached to the tRNA. So this is now the activated amino acid being attached to this tRNA in the 3' end. The amino acid, remember, is attached to the 3' end of the tRNA molecule. This is the 5' end. This is the 3' end. Okay. So we'll now discuss translation. What is step 1? Step 1 is initiation of the protein synthesis. And what is happening during step 1? Look at this step 1 here. The mRNA molecule will align itself on the small subunit of the ribosome. Ribosomes, when they are functional, carrying out protein synthesis, it is composed of two subunits, the large subunit and the small subunit. During initiation of protein synthesis, first, you only have a small subunit involved. The mRNA will align itself on the small subunit so that the initiation codon, which is AUG, okay, will be now at the P site of the small subunit. It is at the ribosomal P site. And then what will happen next? Once the mRNA is being able to align now on the small subunit of the ribosome at the P site, then the tRNA now will start bringing the amino acid as dictated by the first codon AUG. And how will the tRNA know what specific amino acid to bring? Based on the codon AUG, it has an anticodon UAC. And UAC now will enable the tRNA to bring methionine to the P site of the small subunit. So a tRNA with an attached informal methionine will now form a hydrogen bond to be able to attach to the codon. A hydrogen bond will be formed between the anticodon and the codon in the mRNA molecule. And after attachment of the tRNA to the codon, then what will happen? A large ribosomal subunit will bind now to the small subunit, forming what we call the initiation complex. So those are the things that will happen during initiation of protein synthesis. So next, step 2A is elongation of the chain. So first, you have now a tRNA hydrogen bonded to the codon in the mRNA, bringing methionine. I forgot to tell you about this. In bacteria, okay, in the synthesis of the protein in the bacteria, the first amino acid to be brought to the site based on the initiation codon is methionine, and attached to this methionine is N-formyl. But in human beings, it's simply methionine without the N-formyl that will be carried by the tRNA to the small ribosomal subunit. So we are now in step 2A, elongation of the chain. On the P side, you have a tRNA bringing with it methionine. And what will happen? There is now another site we call the A site, to where another tRNA will bind. So if the codon of the mRNA is UUU, the anticodon in the tRNA will be AAA. And a bond will be formed between the three nucleotides, hydrogen bond. And UUU calls for phenylalanine. So that's the way how tRNA will bring the correct amino acid during protein synthesis. So the tRNA, a new tRNA will bind to the A site, and we now have an enzyme we call peptidyl transferase. What is the catalytic activity of this peptidyl transferase? It will now cause formation of a peptide bond. it will cause formation of a peptide bond between phenylalanine, the second amino acid folded by mRNA, and methionine. How? The nitrogen atom of the amino group of phenylalanine will carry out a nucleophilic attack on this carbon atom of the carboxyl group. Okay? On this carbon atom. Forming this one. This is now the peptide bond between methionine and phenylalanine. So once a peptide bond is formed, this tRNA loses its amino acid. This becomes empty. This becomes now empty after a peptide bond is formed. And what will happen once this becomes empty? This will now detach from this codon. And this new tRNA bringing another, so this is now the tRNA containing 2 amino acids, a dipeptide. Okay? So that's the elongation. So what will happen next after peptide bond formation? After peptide bond formation. So this becomes empty. There's tRNA at the P side, while in the E side, you have the tRNA attached to it are 2 amino acids bonded together by a peptide bond. So how will elongation occur? So there will now be translocation. This empty tRNA, where is your empty tRNA? This becomes empty. This will now detach from this codon. This empty tRNA will detach from this codon. Okay? And as you can see, the ribosome moves from the 5' towards the 3' end. So after this becomes empty, what will happen? The one occupying the tRNA containing the 2 amino acids, this one, sorry, because this is another figure. The amino acid here, so if you can still recall, this becomes empty. So you have now at the P side, the tRNA containing phenylalanine and methionine. And then what will happen next? Another tRNA will bind to the E side, bringing valine. So the translocation is simply the tRNA which is being empty, detaching from your AUG, and then another tRNA bringing another amino acid at the A site. And then another peptide bond formation will occur. And the process will repeat until the ribosome reaches the termination codon. So that's translocation. So the P site is occupied by the tRNA with the growing peptide chain, this one. And then another tRNA will now attach to the next codon, bringing another amino acid. And then peptide bond formation will occur between valine and phenylalanine. And then this tRNA will become empty after a bond is formed between phenylalanine and valine. So this will become empty. This will detach. And then another amino acid. And once this will detach, this will transfer to the P site. This tRNA having 3 amino acids will transfer to the P site, and another tRNA will attach to the A site. So that's the process of how the peptide chain will become elongated. Step 3 will now be termination. So the ribosome will keep on moving from one codon to another until it reaches a stop codon. And what will happen during termination? The stop codon does not code for any amino acid. a release factor will bind to the stop codon. A release factor will bind to the stop codon. So there is now binding of the release factor. And what will happen? The bond attaching the carboxyl end of the peptide chain to the tRNA will be cleaved. Again, once the release factor will bind to the stop codon, the bond connecting the carboxyl group to the 3' end of the tRNA will be cleaved, and the peptide chain is now released. The formed peptide chain, polypeptide chain, is now released. And the other thing that will happen during termination, you have different factors. The intrinsic factor, the elongation factor G, bound to GTP that will hydrolyze guanosine triphosphate. This is a ribosomal factor that will now cause dissociation of the complex. This is what we call the mRNA ribosomal complex. Okay? So these different factors, intrinsic factor, elongation factor G, and this RRA factor will now cause dissociation of the mRNA ribosomal complex together with the tRNA in the release factor. They will dissociate from one another. So both the large and the small subunit will also dissociate from each other. So those are the things that will happen during termination. Release of the polypeptide chain and dissociation of the mRNA ribosomal complex. And dissociation of the small and the large ribosomal subunit. So if there is post-translational modification of mRNA, there is also what we call in post-translational modification of the polypeptide chain or protein that has been synthesized. So differentiate between in post-translational modification. When you say co-translational modification, the modification of the protein or the polypeptide chain occurs also during synthesis of the protein molecule. So co-translational modification means the modification of the synthesized protein occurs as it is being synthesized in the ribosome. So again, co-translational modification. The modification of the protein molecule will occur at the same time when it is synthesized in the ribosome. While in post-translational modification, the protein first has to dissociate from the ribosome, and only then will modification occur after the protein has been released from the ribosome. So what is the importance of post-translational modification of the protein or the polypeptide chain? So take note how there are two types of translational modification. If it is of the co-translational type, meaning after the protein is released, it is already functional. After it has undergone co-translational modification. Because the protein has been modified at the same time when it is synthesized. So automatic, when it is released from the ribosome, it is functional already. But a certain type of protein that is modified only after it is released from the ribosome, it is not functional yet. So it will first undergo what we call post-translational modification. And what is the importance of the post-translational modification? It will make the protein functional after it has been modified. So what are the different types of modifications that will occur on the protein molecule after it has been released from the ribosome? One, methyl groups can be attached to certain amino acids in the protein molecule. Methyl groups can be attached, or a phosphoryl group can be attached to certain amino acids in the protein molecule, or a carboxyl group will be attached. A carboxyl group will be attached. So I'll give you one example. Okay. The protein glycogen phosphorylase. One post-translational modification that will occur on the protein, the enzyme glycogen phosphorylase. When it is activated, a phosphoryl group will be attached to the serine side chain. Remember glycogen phosphorylase? Leaving glycogen bonds are glycosidic bonds between glucose molecules in the glycogen. For it to be activated, a phosphoryl group will be attached. That's one type of post-translational modification. Or another type of post-translational modification, if you can still remember, enzymes synthesized in the pancreas, they exist in the zymogen or inactive form. To activate it, after it will enter the duodenum, what did we say? We have the so-called removal of peptide fragments. And the manner of post-translational modification here, there we call it proteolytic activation, removal of peptide fragments. So that's what we call post-translational modification. Modifying proteins by removing fragments, that's what we call proteolytic activation for the enzymes that come in the zymogen form. For the attachment of a carboxyl group, a phosphoryl group, modifying the protein, making it functional. So we are done. We will not take this up next Wednesday of next week, mutation.